If you wanted to show where a protein binds to DNA, you would use a footprinting assay. This would tell you exactly where the protein is sitting on DNA. Begin with a double-stranded piece of DNA labeled at one end. Here's the label, the orange dot. Next, you would bind the protein to the DNA. Then you would digest the DNA protein complex under mild conditions with DNase 1. This is to introduce approximately 1 break per DNA molecule. Next, remove the protein and denature the DNA, yielding the N-labeled fragments shown at center. Notice that the DNase cut the DNA at regular intervals except where the protein was bound. So as you can see, it was cut here, 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 etc. However, there was no cuts here. Finally, electrophorese the labeled fragments and perform autoradiography to detect them. The 3 lanes represent DNA that was bound to 0, 1, and 5 units of protein. The lane with no protein shows a regular ladder of fragments. The lane with 1 unit of protein shows some protection, and the lane with 5 units of protein shows complete protection in the middle. This protected area is called a footprint. It shows where the protein binds to the DNA. Sequencing reactions performed on the same DNA in parallel lanes are usually used. They serve as size markers that show exactly where the protein is bound. If we go to this image over here, this shows an actual experimental result of a DNase footprinting experiment, where, as you can see, with greater increase of protein, a greater footprint is shown, a more clear footprint. A DNase footprinting assay is a great way to tell you where a protein binds to DNA.